All right. Hi, everyone. So, for those of you that are in Mr. Ritchie's class, this is the last video that I'm going to be doing. So, unless, he is out or anything like that, or he needs me to record a video. So, he's going to be starting up his Zoom lessons on Monday. So, Monday, February 1st. Those of you that are my students, I'm going to go through essentially what I'm going to talk about in this video during class. And so, if you just need a refresher, you can watch this video. All right. So some of y'all, in both classes, in Mr. Ritchie's and my class, I've noticed that y'all have already started up the lab. And so I just wanted to go over some instructions just to make sure y'all understand what you're doing. So this lab, investigation mitosis, we're going to be looking at onion root tips. So if you've ever had an onion, you'll notice that the roots, typically are hanging off of one end. So even if you go to the grocery store, if your parents buy onions and they're sitting in your fridge, you'll notice sometimes that there's roots there. And if you let them sit there long enough, the roots are going to grow. So onions are one of the easiest things to grow. That's why, like, if you go to the grocery store and you buy green onions, as long as you only chop off the green part, you can stick that white part in the dirt and you're going to get more onions. So onions are really easy to grow. So for this investigation mitosis, because the roots are actively growing in those onions, then that means that the cells that are in those roots are going through mitosis. So that's what we're going to be looking at. We're going to be looking at onion roots so that way we can see, how many cells are actually going through mitosis. So the beginning of this lab has some introduction. It explains the different parts of an onion root because, of course, they have to be more complicated than what we think they are. And then there's some introductory questions that you get from the intro paragraph. Okay, so right here, when we're talking about our cell cycle, this is going through our different phases just to remind you of, what's going on. And remember, I taught you that hand gesture. So you have the interphase. Okay, so you have a circle. Okay, prophase, where you have the chromosomes, meta in the middle, ana where they're separated, and tela where you have two nuclei. So knowing that, okay, let's go through and we can label these really quick. So my first cell right here, okay, I could see it's a circle, okay, and we're starting with interphase. So I'm going to go ahead and I'm going to type interphase right here. Next, I have chromosomes, and I have a little cartoon up here that shows the chromosomes. So students seem to have the hardest time distinguishing interphase and prophase. Prophase, you're not going to see a line around the nucleus, and you're going to actually start to see little squiggles inside the nucleus. So anytime you see any squiggles, that's prophase, okay, not interphase. All right, that's probably the best way I can describe it. Next, I could see I have chromosomes lined up in the middle. Okay, so I know that's going to be metaphase. Okay. Right here, they're starting to pull apart. Okay, so that's going to be anaphase. Okay. And now, two nuclei. Okay, that's telophase. Okay, so we had inter, pro, meta, ana, tele. Okay, so the hand gesture that I taught you really does work. And my little camera, let me see, I can move my camera. Okay, so this one's telophase. All right. So after telophase, that's when cytokinesis begins. And you can see, because this is a plant cell, that cell plate is starting to form. Okay, so in this little plant cell. All right, so those were some demo slides. Let's keep going. Okay. So here's some more. Okay, so we're going to label the phases again, and now they're out of order. Okay, so I think you could tell which one's interphase, but look down here. Okay, so this one right here is prophase, and you can see there's some little squiggles, there's some little dots, but it's not quite in a circle. Okay, so go ahead, label these cells. Okay, so if you need to reference the previous slide, you can do that. And then there's an opinion question. Which phase do you think the hardest is to identify and why? Okay, and that's completely opinion. So, whatever it is that you think is hardest, that's what you're going to put right here. Next, we need to calculate how long it takes for these cells to go through mitosis. And so typically the total time for a cell cycle in an onion is 720 minutes. That's 12 hours. Okay, and that's why over a period of time, sometimes you will see that, a plant, an onion plant, is actively growing because, 12 hours, if we're talking about a day, then we have two cell cycles going on in a day. All right, so you're going to answer the question, look at these cells, and I know they're really hard to see. Okay, so just go ahead, observe the best you can and take a guess, and then make a prediction. Okay, what phase is longest, which one's the shortest? All right, so right here is where I really wanted to spend some time and, kind of explain what you're doing. Okay, so it says access the cell images in this Google Drive folder and complete the table by counting the number of cells in each phase. Okay, this may be challenging, and some cells you might need to make a decision about. So I would recommend finding a buddy. That way you don't have to do all 5 slides. Okay, so I'm going to go through. I'm actually going to do the first one with y'all. That way I can help y'all out. So Google Drive folder. Okay, so here's my first slide. Okay, so there's a couple things looking here. Okay, so first off, looking at this slide, I can see some cells that are going through mitosis. Like I could see here's an anaphase, here's a telophase, there's a prophase. So I know we have, some that are very obviously in mitosis. If I look up here, okay, so right up here, some of my cells look like they're overlapping. Okay, so like I can see there's some cells behind this layer of cells right here, and the same thing for right here. Okay, so if I have cells that are kind of in the background that I can't really tell what they are, I'm not going to count them. Okay, so like these right here, these dark kind of, well, not dark, they're kind of light splotches. I can't tell what those are, so I'm not going to count them. Okay, and remember, you don't have to be perfect. Okay, you just kind of need to be generally correct. All right. Also, these cells over here at the side, again, I can't really tell what they are, and the ones in the, in the corners and stuff like that, can't really tell what they are. This one I could tell is a metaphase, so I will count that one. But I'm not going to count the rest of these. Like if it's obvious, like this dark one right here, okay, I know those are interphase, I'll count those. But like stuff like this over here, I won't count. This one up here, I won't count. These guys right here, I won't count. Okay, so you're just going to do the best that you can. Try to count the majority of the cells here. And as long as you're pretty close, you should be fine. All right, so what I'm going to go through, okay, I'm going to go ahead and count. I'll go ahead and start with prophase. Okay, so I'm going to go through and count my prophase cells and go metaphase and so on. And so my last ones that I will count will be interphase. Okay, so just glancing at this, okay, so I'm just going to go line by line. Okay, so this one right here, prophase, so 1. So another prophase, 2. And I'm going pretty fast. Okay, but it's because I've done this before. So another prophase, so 3. So another one right here, 4. Okay, so I see 4. This one might be 1. We can count it. We can say that's 5. Okay, so 5 prophase. Okay, so I'm going to come over here. Okay, so prophase, I'm going to put 5. Okay, so next I'm going to do metaphase. Okay, metaphase are easy. There's one right here. 1. Make sure I don't miss one. I think there's just 1. I don't see another one. Okay, so I'm just going to put 1. Anaphase is when they're pulling apart. So let's see here. Anaphase. Okay, so there's 1 anaphase. Yep, there's just 1 anaphase. All right, now telophase, where I have 2. Okay, so let's see here. Okay, there's 1 right there. I think I just have 1. Okay, so 1 right there. All right, so now I'm going to count my interphase. That's a lot. Okay, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36. Let's see here, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 48, 49, 50, 51, 52, 53, 54, 55, 56, 57, 58, 59, 60, 61, 62, 63, 64, 65. Okay, so I'm going to say 65. So you may have noticed that, some I counted, some I didn't. For interphase, it's okay. It's okay to have, a very rough guesstimate. Okay, so you just know that you have majority interphase. So these are going to be my numbers: 65 interphase, 5 prophase, 1 meta, 1 ana, and 1 tele. Okay, so now you're going to go through and you're going to do slides 2 through 5. Okay, so and some of these are easier to see than others. Okay, so like this one's pretty easy, and you can see there's a big white space over here because this was the edge of a root. Okay, so there's this one. There's quite a few metaphase in that one. That one. And this one. I think I did the hardest one for y'all. Okay, so you'll go through slides 2 through 5, and you will go through and count those cells. So once you do that, you're going to figure out what your total is. So you'll have to add all those cells together in the row. And then you're going to use your formula to figure out the time. Okay, so for this time, you're going to use this formula that's right down here. So your number of cells in a specific phase divided by your total number of cells counted. Okay, you're going to multiply that by 720 minutes, and that's your time for your specific phase. Okay, so that's how you're going to calculate time. And then from there, you'll figure out your percentage. Okay, so you're going to take time divided by 720 and multiply it by 100. So right here, you'll use your data from the previous table. Okay, so what's the longest phase? So are other phases close or equal? Like if we see over here, okay, meta, ana, and tele, those are pretty equal. Let's see here. Okay, so you were asked which one would be most difficult. Okay, so were you correct in thinking that was most difficult? So now, okay, so that was for a plant cell. We're going to look at an animal cell. So this is a fish blastula. Okay, so we use fish blastula because they're actually the easiest to see. And you can tell right here, this is still pretty difficult. Okay, so it's actually really, really hard. So we actually don't do this if you're in the classroom just because it's so hard to see. All right, so if you're looking at the blastula, how is it different from that onion stem? What phase of mitosis can you find on the slide? So you're going to move this circle around and you're going to try to figure out one of the phases. Okay, so you just need to do one. And how do the number of cells in interphase compare to that of the onion? So for interphase, you can see here's one that has a clear nucleus. You can see there's that circle there. Okay, here's another one. There's another one. There's another one. Okay, so just generally looking at it, is it equal to having the same number of cells or same percentage of cells in interphase, or do you have more cells in mitosis than you did for your onion cell? Okay. All right, so right here, okay, after that, so how can a cell that is fixed, okay, be used to estimate the time spent in the phases of mitosis? And explain your process you use to determine the times for each phase. Okay, so we had a formula. Okay, so the formula did help. And then also, even though the slide is fixed, we could still see cells, that were going through mitosis. Okay, so you're going to go ahead, write your final conclusion here, and then after that, you will submit it. So I know this is a long lab. We're going to give you 2 days to do this lab. Okay, so we're going to give you Friday, we're going to give you Monday. Those of you that are in Mr. Ritchie's class, he is going to be helping you on Monday, so don't get scared if there's something that you don't know how to do. Just, make sure you write down the questions so that way you can ask him. Okay, so I'm also available for questions. So when you start this lab up, if you don't understand something, you can definitely send me an email. All right, so I hope you have a great day, and I will see y'all later.